As you know, my name's Sam Cobb. I'm the CEO, founding CEO of AdAlta. Been with AdAlta nearly 12 years now. And my background is science, law, and commercialising science. So AdAlta, we're listed on the ASX. We've got a novel platform which we call the iBody. We've used that platform technology to identify our lead candidate, AD-214, which we've been talking about today. The focus of AD-214 is primarily to move it to the clinic, complete a phase 1 study, and look at idiopathic pulmonary fibrosis. We have our orphan drug designation in the US for the treatment of IPF. What we haven't really talked about today is the commercial opportunity, so the business opportunity. So what we're seeing, and we heard from the panel just now that the traditional big pharma come in at the end of a phase 2. What we're really seeing in idiopathic pulmonary fibrosis is earlier commercialisation transactions, and mainly driven by, Steve went through the list of clinical trials. There's 2 drugs that are approved, there's a few drugs in the clinic, but not that many. So they tend to be bought up by the bigger companies. BMS, where Robert had worked, has been quite active in that space, and I can go through some of those examples. As you've seen today, we've got a great team with Dallas heading up manufacturing, Mick, who you haven't heard from today, but our Chief Scientific Officer, and with the Scientific Advisory Board. Really our focus is to take AD-214 to the end of that phase 1 where we see a lot of those transactions. At the end of the December quarter, we had 5.31 million in cash, and we have strong institutional support. So Hugh McNally gave a presentation this morning about how hard this sector is, but we do see a lot of support from institutional investors. So our code is 1AD. A couple of days ago, our share price was 27 cents. We have a market cap of 31 million. The share price in the last 12 months has gone from 20 cents to 40 cents. Our major investor is UR Capital, so UR, represented by Liddy, McCall and James Williams, have also been here today. They have about 45%. Platinum Asset Management aren't here, but a major institutional investor in AdAlta has 9%. Leon, who was here, City Castle, has about 5%. And then with Hugh as well investing, as well as a high net worth called Peter Mewers. So that we have a quite a tight shareholding, with the top 20 shareholders holding 80% of the company. Very strong institutional support, with about 60% institutional investment into the company. So I'm the founding CEO of AdAlta. Also here today, earlier today, was Paul McClellan, our chairman. Hopefully you got a chance to say hi to him. Robert Peach, a director of AdAlta, significant antibody development experience, what we've heard in terms of SC fusions, taking products right from the start all the way through to the clinic. John Chiplin's based in the UK, again also a lot of antibody experience, sold the company that he was working with Adarana Therapeutics to Cephalon. And then James and Liddy, both from UR Capital, again have sold multiple companies and taken multiple products through to FDA approval. So you've met everyone today from our Scientific Advisory Board, including our CSO Mick Foley. I probably don't need to go back into their backgrounds in detail, but just to say that they provide me and the AdAlta team a significant amount of expertise in drug development, which is very, very helpful. We're developing our lead candidate AD-214 in the area of IPF. We've heard from a clinician today, Glenn Westall, about the disease, the 2 drugs that are available to patients. Steve's given a great background about the drugs that are currently in the trials. Not one of those drugs is focused on CXCR4, as we've heard. And we've also heard from a patient who really has spoken to the need for new drugs, that the average life is about 3 to 4 years. The 2 available drugs work in about 30% of patients. Side effects, you go off these. And there's from a market point of view, these drugs sell about $1 billion per annum. And so there's from a pharma point of view, a real market opportunity. From a patient, a real unmet medical need, and a real need to develop new drugs that are better than what's already out there for patients. So our drug AD-214 binds to a target called CXCR4. We've also heard about that and how what we've seen with Glenn and his team and other researchers is that we have the target is only upregulated in disease. So we see an expression only in diseased tissue, in the diseased lung, not in a normal patient. And this is critical from a mechanism and a point of view that we're different to other products. Our eye-body will bind to the disease target and have an effect, and we've seen this in a number of different animal models, so having both anti-fibrotic and anti-inflammatory effects. We've got our orphan drug designation, and last year published in Nature Scientific Reports some of that data, so looking at both animal models in the lung but also human tissue with Glenn on that paper as well. So AD-214, this is some of the animal data in the lung, and you can see on the left, as Glenn spoke about, those air pockets. In the middle, the diseased lung, so you get this build-up of this protein called collagen caused by those fibrotic cells. The areas in blue that really block all of those airways. And you see on the far right here, the actual picture of the diseased lung treated with our eye-body. So a significant improvement in the architecture or the look of that picture of the lung. What we've also shown, and I think this has also come up during the day, not just one animal model. We've shown it in the lung and animal models of the eye, so age-related macular degeneration, in the liver, the skin and kidney. And in all of these animal models, we're seeing both anti-fibrotic and anti-inflammatory effects. So where you see some challenges potentially with those models, we've looked at both human tissue as well as these animal models and shown that across all of these, we see the same effect, an anti-fibrotic effect. One of the keys, the differences between the commercialisation activities in the area of IPF is the early transactions. Fibrosis assets, because of the unmet medical need and the large pharma and the market opportunity, we see a number of acquisitions. And so a number of these, this is all of the idiopathic pulmonary fibrosis deals since 2011. All of the deals that have happened in this space have happened around about the end of phase 1, with the exception being pirfenidone being acquired by Roche, which was acquired at the end of phase 3. So you see the average upfront payment being around 100 million at the end of phase 1, and between 300 and 400 million in milestone payments being paid. The more recent transaction there was a 10 million upfront. This was for USA rights only. So you see the majority of these deals are for worldwide rights to the compound for IPF, for the treatment of IPF. So Dallas earlier today gave us an update on how we're going with AD-214, and on Tuesday we made the announcement that we're on track. This is critical for AdAlta to meet milestones moving forward, to get into our non-human primate studies, to get into the clinic. We really need to be able to manufacture this with a process and clearly be able to articulate this to the regulatory agencies. We also want to make sure that what we put into people is safe. So we've almost completed, as Dallas mentioned, the cell line development. We've spoken about a 3 gram per litre at the moment yield through that expression system. And at the moment we're optimising how we're expressing that protein so that we can get the most AD-214 possible, and then optimising a process for purification and how we store that, formulate it in a formulation buffer. So all of this going to plan. We'll have our materials for our big 4-week toxicology study in beginning of July this year. We've got a couple of non-human primate studies that we'll look at between now and then. These are really focused around the half-life of the eye-body AD-214 and looking for these markers, biomarkers that we've already spoken about. So how long does the eye-body last and what effect does it have in a monkey? We'll take those and incorporate those readouts into our 4-week toxicology study in the second half of this year, and then move into a standard healthy volunteer study in January 2020. So completing the safety evaluation of AD-214 the first half of next year. In parallel to that, we haven't really touched on today, but we've got the eye-body platform. So where AD-214 came from was the library of iBodies that we have access to, the screening capability of identifying new products for new treatment, for new therapies, also sitting behind AdAlta's lead candidate AD-214. Our goal also over the next 12 months is really to publish in peer literature the results in the various animal models of AD-214 and AdAlta's lead iBody candidates, because we know that pharma are constantly watching this, and that peer validation in journals is also key. And as we've mentioned, I'm out there talking to pretty much everybody about what we're doing. So the significant achievements that we've had over the last 12 months is really orphan drug designation, completing and showing a broad antifibrotic effect in not just one animal model, multiple animal models, publishing the data, and completing some initial studies to show that AD-214 is safe, but also the compound prior to that, AD-114. So putting together a package of data which demonstrates not only the efficacy but the safety of the compound so that we can move into the clinic. We have the patent for AD-214 granted in Australia, and the manufacturing, as mentioned, is on track. So the business model for AdAlta is to license out AD-214 where we can get the most value for shareholders. We've also got a platform technology where we can go after those difficult targets that other people are looking at, but we've also got our own list where we can say the iBody technology is well suited to difficult drug targets. We're looking at doing that with our own in-house pipeline development, but also my focus is going out and talking to pharma and biotech companies and how we can use that technology and be funded to do the R&D for other companies. So looking for partnerships where we can apply the iBody platform. So in terms of financial benchmarks, we see the company positioned in a number of different areas. So the lead candidate being positioned for a licensing deal where we see the average upfront payment about 100 million and between 300 and 400 million in milestone payments. This next generation antibody, or the iBody, we see multiple R&D, so research and development contracts and partnerships being put in place where pharma are coming to companies like AdAlta and paying for research projects. As an example, AdLinks did a project in July 17 with Sanofi. They paid 31 million euros up front, and they paid up to 2.4 billion in milestone payments, but as of last year were acquired. AdLinks actually was acquired by Sanofi with some competitive tension from some other partners they also had. And then we see the area of G protein coupled receptors, so traditionally difficult to drug targets, and we see the iBody and its long loop, which we haven't really spoken about, getting into those targets that have been traditionally very difficult to drug. And we've seen a couple of companies, so AdLinks again, a very similar technology to AdAlta's, but ours being human-based, a human protein, we can do very similar things and getting in and doing that. And we saw again AdLinks doing a deal with Boehringer where Boehringer paid them to do a research project for 8 million up front. So we're out there talking to parties about how we can partner with them for research projects. So expected news flow over the next 6 months, we're looking at completing the manufacturing, publishing the data, and some preliminary PK data, so half-life data in from non-human primates. And then the second half of this year, real key for us is completing our 4-week toxicology studies, starting to talk to the FDA about how we'll move into the clinical trials in the US, and then in the first half of next year, getting into the clinic with the phase 1 study. So our cash position as of December last year was 5.3 million. We've got a lead compound with significant preclinical validation in a number of areas of fibrosis. Our focus is to take that forward for the treatment of idiopathic pulmonary fibrosis in the first instance. Our manufacturing's on track. We've got the platform underpinning and allowing for future product development, but also partnerships. And as you've seen today, we've got an experienced team behind us, enabling us to get that there, both product and the platform. So thank you all for attending today, and I'm happy to answer any questions.
What are the beating CXCR4 binders that are on the market? And how does iBody compare to them?
Yeah, so there is an approved CXCR4 small molecule. It's an approved drug it was originally developed for HIV treatment, but they found that it was actually quite toxic, cardiac toxic, so you actually had cardiac problems with the drug as a small molecule. It had a lot of off-target effects, we've spoken about those. It's actually been approved for a number of years now for a single dose in patients for stem cell mobilisation. So they give it to patients before they get their chemotherapy. But because of the cardiac toxic side effects, it can't be given long term. There are a few other drugs in development, and they're all being developed for stem cell mobilisation. They're all being developed in the area of cancer. So the key difference between our drug and those drugs that are currently in the clinic for stem cell mobilisation is that we've used that long loop of the iBody to really modulate or activate or block pathways quite differently to that small molecule. So we're actually having a very different activity compared to those. So we do not mobilise stem cells. We have quite a different activity when you look at some of the work that we've done with human tissue. As an example, we found that that small molecule that's been approved has no activity with human tissue, zero activity at all. Whereas AD-214 and our previous molecule AD-114 actually could stop the movement of these fibroblast or fibrotic cells, and also stop those cells from producing that collagen that we saw in the animal model. So while AMD-3100 works in an animal model, it doesn't work in human tissue, and our molecule does. So there's some real key differences between what's already being developed, what's approved for CXCR4 and AD-214. So we see it really as a key advantage moving forward. So it's Mark.
What sort of data do you expect to get out of the phase 1 study, and on the back of that, if it's positive, will you aim to do a deal?
So the phase 1 study we've got at the moment is looking at healthy volunteers, so a single ascending dose, and then a multiple ascending dose, so multiple doses all in healthy volunteers. So from that, we're looking at what is the PK in a healthy volunteer population, and is there any change in any of these biomarkers that we're still working on. One of the other studies we're looking at is a phase 1B. Sorry, if I take a step back, the phase 1 safety is really where we see a lot of these other transactions happening. We're also looking at the moment with the scientific advisory at HELP and clinicians like Glenn, and mapping out what a 1B would look like. So a smaller group of patients, how does the PK or the half-life change in the patient population? Because from the testing we've already done, we see that a normal patient doesn't have CXCR4. So when you have a disease patient, does the half-life change for that molecule? And are there any preliminary biomarkers that we can get early readouts? So we see a 1B really key for partnering for us, in that we'll be able to, in the patient population, demonstrate what the half-life is and if there's any potential early readouts with biomarkers. So not expecting the longer-term forced vital capacity or efficacy markers, but looking at short-term readouts that we possibly could get. So they're the key, really, phase 1 studies.
So the biomarker development, is that underway?
We're doing it at the moment, yeah. It's ongoing. Yeah. Peter?
What is your cash burn?
Dallas is a great spender on manufacturing. I think this quarter we'll spend about $3 million. So last quarter I think we spent $1.5 million, and that really is reflective of not just going through cell line development, but doing upstream, downstream formulation all in parallel. So we're burning through quickly, but we're getting to the point where we're saying we want to get that material and we want it as fast as possible. So we will burn through $3 million this quarter. So we did announce in our 4C that we will need to raise money. And the board is obviously looking at various options. So any other questions? No? Well, I just want to thank everybody once again for attending. Always happy to answer any questions that you might have, and thanks for coming. Look forward to updating you on an ongoing basis with our progress. Thank you.